Hello, everyone. Good morning. First of all, I would like to thank you all for joining for our spring seminar series today. As all of you know, I'm Avinash Shrestha. I'm currently the Vice President of the Graduate Council. And it's my pleasure to introduce Arjun today. So, Mr. Arjun Ojha, he completed his master's degree in plant biotechnology from West Virginia State University in 2019. And right after that, he joined in Patil Lab in Isaac from 2022 and has been working since then under the supervision of Dr. Gunbant Patil. And as a graduate student, he has achieved numerous awards and recognitions. And I'm not going to mention all of them, but one of them includes the prestigious Helen David Jones Fellowship. And as a PhD candidate, he is currently working on two major projects, which he is going to present today under the title of Understanding Single Cell Regeneration Pathway and Development of Tissue Culture-Free Regeneration and Gene Editing Platforms in Crops. And with that, I would like to hand the mic to Arjun and let him proceed with his presentation. Thank you.
Thank you so much, Avinash, for the introduction. Good afternoon, everyone. Today, I'll be presenting about my two PhD projects. One of them is the development of tissue culture-free gene editing and regeneration platform in crops. And the second one is understanding the molecular mechanisms underlying somatic cell regeneration in plants. So these are my two major PhD projects, and I'll be walking through you all through these two projects today. Starting with the first project, the development of tissue culture-free gene editing and regeneration platform in crops. Genome editing technology like CRISPR-Cas9 is quite familiar now. It has been developed in 2012, and since then it has been profoundly used in plant engineering research, both in applied and small-scale researches. However, this genome editing technology like CRISPR-Cas9 was ever since then it was developed. It evolved with several challenges. Two major challenges in plant genetic engineering are the transformation system. Several plant species are recalcitrant to transformation, and even if we can transform those plants, the gene editing itself is a major bottleneck. Not only these major bottlenecks, there are several other limitations to plant genetic engineering, like skills. So a high skill is required for the genome editing technologies. Similarly, time. Time is one of the major limitations, I would say, because conventional breeding technologies take several years to get progeny plants with improved trait or genome edited or transgenesis, and also it is highly cost. It's not cost effective. It costs a lot of money, and also genotype dependency. Genotype dependency is another thing. A lot of plant species, very few plant species can be transformed and gene edited or genetically modified. So through my research, I'm trying to work on these limitations of plant genetic engineering, thereby increasing the efficiency, skill, time, cost, and genotype independency. If we look at the example of a cotton transformation, there are only few genotypes of cotton, which is cocker, can be transformed, and the transformation frequency is very low, which is like 0.4 to 2.2%, and takes several months to regenerate into a next generation. Similarly, if we look at the sorghum transformation, which is another important crop, it takes anywhere between 9 to 12 months to get the progeny plants and harvest the mature grain. So these are here in this slide. I'm trying to say, so the different transformation methods. Transformation of foreign gene into a plant can be done through biological delivery method, which is agrobacterium mediated delivery. We can also transform them by bialistic, which is physical delivery, and then chemical or electrical. So we use protoplasts in this case and transform, and then go through the tissue culture intermediate and regenerate whole plants. And if you look at the common thing in all different transformation methods, they all go through a callus formation, which is a prerequisite for the regeneration of the whole plant, and it goes through. It has to go through in vitro regeneration processes where there is interplay between different hormones and regeneration of the plants. So this is the major bottleneck that I'm trying to address in plant genome editing. Tissue culture takes a lot of time. It is a lengthy process. It requires a lot of skills. It also costs a lot. So we are trying to bypass all these stages through a different method. And depending upon the type of plant species you are considering to do the transformation, genome editing, and regeneration, it takes anywhere between 4 to 12 months for the regeneration of the plant with a new trait. So to avoid this or to bypass this tedious method of transformation and regeneration, we are trying to develop a direct delivery system where we use agrobacterium as a mode of delivery of our gene editing reagents and transform directly into the plant, which are grown in the greenhouse or growth chamber. Basically, we make agrobacterium and then we transform it into the plant, and we expect to get a new plant which is genetically modified. Either it is a gene edited or transgenic at the site where we inject our construct. And this is not so the direct delivery system has been tried by different other labs previously. One of the labs tried this in tobacco, and they were able to regenerate the gene edited, which is the white plant here, from the plant. This is in-planter transformation, which is a direct delivery system. And there is another paper which was published in 2012. They also tried the direct delivery system. So the question you may ask is, why are you trying the same thing which has been already done by different labs and different people? And the answer to that is genome editing technology is always evolving, and we are trying to enhance the efficiency of genome editing. Genome editing frequency and regeneration frequency are still very low in these technologies, these projects they have done, and the project and the method that I'm employing in my project is completely different from what they have done. Though the method of delivery is same, but it is completely novel compared to this. And when I walk you through different slides, I will compare these studies with my studies to say that my study performs better compared to what they have done previously. So it is not novel in a sense, but it is novel because everything is novel in my pipeline. Basically, regeneration process occurs in two different stages. One is differentiation, where the cells proliferate vigorously, and then they de-differentiate to form the new plants from the differentiated cells. So there is a two-stage process for the regeneration of the plants. And for the regeneration of the plants, to de-differentiate, there is a stem cell activity where genes like WuSCHEL and clavata come into play. And these genes are also called morphogenic regulators or developmental regulators. And I'm also applying these developmental regulators to enhance regeneration in my project. Basically, my construct looks like this. It has a CRISPR reagent, it has an inducer that helps in differentiation process, and it has DR. DR means developmental regulators, which are genes involved in stem cell activity here in pseudoapical meristems, and multiple DRs there, which induces the formation of differentiation. DRs de-differentiates those differentiated cells, and there is also a Ruby gene, which is a visible report gene. Okay, so the inducer gene, which is here, is guided by 35S promoter, and that will induce the promoter for the developmental regulator. So this is how it works. So this part will differentiate the cells, and then those differentiated cells are induced to regenerate with the help of developmental regulators. So after I made those constructs, I wanted to check whether they are actually working or not before I move on to injecting and transforming directly in planta. So I have generated two different constructs. That is a Ruby gene. There is nothing in there, but only Ruby gene under this promoter, CMOLCB promoter, and my inducer gene under 35S promoter. So I used these two genes and did a conventional tissue culture taking tobacco explants. And as you can see here, with only Ruby, we can just see the expression of Ruby, the red region there. There is no proliferation or differentiation of the cells. But when there is an inducer gene under 35S promoter, you can see a massive callus formation. And callus formation, like I said, is the prerequisite for the regeneration of a new plant. So 35S promoter under that is guiding the inducer gene produces a lot of calluses, and I measured the area of these calluses, and PAK50 is that one, and 73 is this construct. So as you can see there, the area of callus is significantly larger with the inducer gene compared to just an empty construct. So next thing is, okay, so half of my construct is working. So the next thing is to check whether the other half of the construct is working or not, which is helping in regeneration. So the developmental regulator genes. So I further subjected these explants with a different, so I did a different experiment with the whole construct that has this gene plus the developmental regulators, and the tobacco explant regenerated without the supplement of the hormones. Basically, in vitro condition or in tissue culture pipeline, we supply the media with the hormones, auxin and cytokinin. Auxin helps in the callus formation, and cytokinin helps in the shoot formation. But without the application of exogenous hormones, growth regulating hormones, because of these developmental regulators and inducer gene, I was able to regenerate the plant from the explants that I have used. So this shows that both my inducer gene and the developmental regulators are working. So it's now time to check whether it's going to work in the plant direct delivery. So again, this is my construct. It has different parts in gene editing. CRISPR reagent is for gene editing that will target a gene called PDS. PDS is a phytoin desaturase gene, and it is involved in photosynthetic pathway. If we knock that gene out, the plant looks completely white phenotype. Biallelic mutation of this gene will look like an albino phenotype. Similarly, I have inducer and DRs for regeneration, and I also have a visible reporter Ruby gene. So how am I going to do this? Is grow a plant, tobacco plant in soil. When it is about a month old, initially I wanted to try 3 different strategies for injecting the particular construct into the plant. First strategy is to inject in 3 different locations in the plant. On the top near the shoot apical meristem, in the middle, and in the bottom. So the injection site is always an internode where there is no way a plant can regenerate from there. It's between 2 nodes. That is where I was planning to inject. It's not in the it's completely in the non-meristematic region of the plant. Second strategy was to inject only on the top of the plant, where near the shoot apical meristem. And third strategy was to completely remove the apical dominance, cut the plant, and inject the construct there. And the expectation was wherever I inject my construct, wherever I inject this construct, a new plant will regenerate from there. That was the main idea. The red plant you can see here is a transgenic, either transgenic or gene edited plant. And so I injected in 3 different spots, one spot in the top. And after injecting with the agrobacterium, I put a cotton plug there to prepare to give a conducive environment for agrobacteria to infect the plant and transfer the construct into the plant genome and modify the genome. So the cotton plug is basically soaked in agrobacterium. And after this, I transfer these plants into, incubate these plants in dark for 3 days, which we also call as co-cultivation period. And then after 3 days, they will go back into the normal growth chamber where there is like 22 degrees Celsius, 16 hours light, and 8 hours of dark. And what we expect to see is now after we put them into the growth chamber, after infection and co-cultivation, we expect to see a new plant growing from these regions. So unfortunately, when I injected in 3 different locations, what happened was there was only the healing of the wound and there was no regeneration of the plant, or not even the differentiation or callus formation in the site of injection. When it was injected in 3 different locations, there was a massive callus formation, as you can see here, but still there is no regeneration. But when it was injected on the top, after removing the pseudoapical dominance, we started seeing the regeneration of de novo shoots there. And after this, we thought that, okay, only the third strategy is working, and we started following only the third strategy afterwards. All right, so after we have this pilot project on how different strategies could work, then I started working on this technology, only third strategy, and the third strategy could give me both gene edited and transgenic shoots at the site of injection. As you can see here, the white the white leaf here coming from this site where I was injected, as you can see here, is the biallelic mutation of that PDS gene. So knocking out that gene gives the white phenotype. The red plant here are a transgenic plant expressing the ruby gene. The ruby gene will give the red color to the transgenic plant, which signifies they are transgenic, and the green plants are wild type plants. So gene editing and transgenesis we can see here in the same picture, and like I said previously, the gene editing efficiency in my experiment was about 12%, whereas in the previous studies where which was done in 2020, they have only 5% gene editing efficiency. So the efficiency of gene editing increased almost more than twice compared to the previous studies. Similarly, induction of genetically modified or transgenesis. So these pictures here shows transgenesis. All the new branches, the red branches here shows the transgenic branch that is emerging, that is coming out of the point or the site where I have injected my construct. They have ruby gene expressed in all the branches. And again, comparing to the previous studies, in this study, I have transgenesis about 35%, whereas the transgenesis in the previous studies was 20% and 7% respectively. So both gene editing efficiency and transgenesis has been increased in this study. Now, while working with the genes like developmental regulators, we expect to get some developmental abnormalities in the plant. As you can see here, this is a normal tobacco plant, and these are all the different plants that has abnormal unusual morphology. This is the red you can see here is a transgenic ruby branches that are abnormal. Here you can see a bushy plant. Some of the leaves are semi-albino, while some of the leaves are completely white. Those are gene edited, and this is normal transgenic, but it's not showing any color because this has a different reporter gene there. So because of the pleiotropic effect of the developmental regulators, we expect to see abnormal phenotypes, but the frequency of abnormal phenotype is significantly reduced in my study compared to what other people have shown before. So it's only 4% in my study, but it was 42 and 22% in the previous studies. So we don't expect to get unusual phenotype because they don't flower and they cannot produce the seeds that could pass to a second generation. So it's not a good thing to get an abnormal phenotype, but it happens because of the developmental regulators. Now, the transgenesis has been transmitted to next generation. As you can see here, the branch, red branch is a transgenic branch that produced red flowers and also seeds, and those seeds were all transgenic. And when they were young, they look red, but when they mature, they are green. But the flowers you can see are all transgenic. So compared to the wild type plant, these plants are shorter. but they are transgenic. And I confirmed the transgenesis by PCR here. Here I have done the Ruby gene amplification to check the transgenesis of in the second generation plant. Um, and interestingly, the second generation plants produce more number of branches and more flowers. So this is very This was very interesting, and I didn't expect that it would happen. This could happen, but the second generation plants produce a lot of seeds, a lot of flowers, and several branches. Though the height of the plant was reduced, but number of branches were increased. So all in all, we have developed a robust gene editing and regeneration technology by bypassing a tissue culture. This can be done in greenhouse. You grow your plants in the greenhouse, and then when they are at a particular age, you can just take your construct and inject, and you could get regeneration of both gene edited and transgenic plants. So conventional transformation regeneration takes up to 12 weeks. And direct shoot induction took about 8 weeks for regeneration in that paper, which was published in Nature Biotech, and another paper which was published in PBJ. They were able to regenerate in 6 weeks. But in my construct, with the help of my construct that has inducer that induces the expression of the developmental regulators, I could regenerate a new meristem within 3 weeks. So that has significantly reduced significantly reduced the time. Now, this has been successfully tested in tobacco. Now, what is the next? Next step is to test whether it's going to work in some crops. So before moving on to recalcitrant crop, I tried to check whether it's going to work in other plant, a close relative of tobacco, and I tried in tomato. And as you can see here in this picture, where you can see a differentiation of the cells at the site of injection, the red callus formation, and then the regeneration. So it works well in tomato as well. So these are the different tomato plants that regenerated Ruby positive plants. Ruby positive means they are all transgenic. And the transgenesis efficiency in tomato was about almost close to 10%, 9 point something percentage in tomato. So it is low in compared to tobacco, but it's still a good efficiency of regeneration in tomato. Next, I was testing this in soybean. I'm still testing it in soybean. So far, I haven't been able to get any regeneration in soybean, but you can see the differentiation of the cells here. There's a huge callus formation there, and some of the transgenic callus also, as you can see there, the red callus. But I haven't yet seen the transgenic plant regenerated from the site of injection. So what that means is that there is accelerated differentiation. Half of my construct is working really well. It gives massive cell mass, but it is not regenerating, meaning that I may need to try different combinations of different developmental regulators to induce de novo shoot formation in recalcitrant species like soybean. I'm also checking this, testing this technology in other recalcitrant crop species like chili. So we have worked on the scale, we have worked massively improved the timing, and also on the cost, and also we are working on the genotype dependency. So far we have tested in tomato, and we are still testing in more crops. So in conclusion, the synthetic cascade is working really well in tobacco and tomato. We have achieved higher differentiation activity with the enhancer gene, and the ectopic expression of these developmental regulators improves transformation and regeneration efficiency in both the crops, tobacco and tomato. Within 3 weeks, we were able to see the regeneration of de novo meristems. And we are testing this system in the recalcitrant species, like I mentioned, in soybean and also in chili pepper. Now, with this, I concluded the first project, and I'm moving on to the second project, which is understanding the regeneration of somatic cells like protoplast. What are the molecular clues that are involved in single cell regeneration, taking tobacco protoplast as my um, taking tobacco protoplast. So basically, a tobacco protoplast regenerates from, if we count from day 0, it will regenerate in 9 weeks by navigating different developmental stages. And I'll walk you through this. So starting with the background, so we know that plant cell can regenerate. Plant cells are totipotent and they can regenerate into an entire fertile plant. Basically, there are different triggers to plant regeneration, and first one is in vivo, which is in nature. External stimuli induce reprogramming and regeneration. So external stimulus like season or other stress stimuli like wounding leads to regeneration of the new plants in vivo. Whereas in vitro or in tissue culture, in laboratory, hormone-induced reprogramming leads to regeneration of the plants. As you can see here, this experiment was done in 1957 by Skug and Miller, where they used different combinations of auxin and cytokinin to regenerate plants, and plants also regenerates in nature. The question is, how does a single somatic cell or a protoplast regenerates into a whole plant is still not very well known. So there are several intriguing questions in plant cell biology that are still not known very well. One of them is like, how does a single somatic cell become the whole plant? How do they reactivate and re-enter into the cell cycle? And what is the mechanism that relocates the nucleus into correct position, thereby deforming fully expanded vacuole? And how do the cells lose chloroplast to acquire new cell fate? And why is there genotype dependency in protoplast regeneration in plants? So these are the several different intriguing questions that has not been answered yet. And the only answer to all these questions is the molecular mechanisms involved in somatic cell regeneration. So we need to understand how plant cells, what are the molecular mechanisms that is required for the regeneration of the somatic cells. So basically, a protoplast is a cell without a cell wall. We can isolate protoplast from, for example, leaf, which has several cells there. We digest them with the enzymes, and then we release the protoplast, thereby digesting the cell wall, and we get the protoplast. And these protoplasts have a remarkable developmental plasticity, and it can regenerate into a whole plant. There are different sources of protoplast. We can isolate protoplast from almost any kind of plant tissues, for example, immature seeds, leaves, roots, cotyledon, hypocotyl flower, and callus. So these are different sources of protoplast that we can isolate. And also we can isolate protoplast from leaf, callus, and root, and regeneration frequency may differ if we isolate protoplast from different source. Basically, there are two criteria in successful protoplast regeneration. The first one is the organ you wanted to consider isolating protoplast. What kind of organ you want to take? Do you want to take a leaf or a callus or a root or a cotyledon or whatever organ you wanted to consider? The second thing is, whatever organ you consider, the types of cells within that organ is very important. What kind of cells are there within that organ you pick? For example, if you pick a leaf, what kind of cells are there? Are they competent to regenerate? That is more important. So while picking the organs for the regeneration, even if we can isolate protoplast from all the different types of organs, they may not regenerate equally, and also they may not regenerate equally when it comes to different species. For example, we say soybean is highly recalcitrant regeneration compared to tobacco or carrot. So there must be a reason why soybean does not, or sorghum does not regenerate as good as tobacco or carrot. So different species and regenerates differently. One of the reasons, so regeneration, what it matters, like I said in my previous, before, the type of organ you wanted to consider is very important for a single cell or a plant to regenerate. For taking an example of tobacco and sorghum or maize, we usually say that tobacco is very easy to regenerate. It's a model organism; it regenerates fairly well. But when it comes to maize or sorghum, we say that it doesn't regenerate. It's hard to transform. It takes several months to regenerate, so and so. And the reason behind that is the type of cells again within the type of explant you want to take. For example, if you wanted to take leaf sample for both the plant species and isolate protoplasts from these plant species, uh, the cells that are competent to regenerate are not distributed equally. For example, all the cells in this leaf, tobacco leaves are fairly, they are all highly differentiated, but and also they can regenerate fairly well. But in case of monocots like sorghum or maize, the competent cells, regeneration competent cells are confined to these area, lower base of the leaf blade, and also in very young seedlings. So say for example, we take the whole leaf from this sorghum and whole leaf from tobacco, and we isolated 1 million cells, for example. If we want to regenerate, the regenerate competent cells in both the uh, the regeneration competent cells in these two plant species are different. Uh, Almost 100% of these cells may regenerate. They have regeneration competence, but only few cells that are coming out of these locations are only regeneration competent. So it matters what kind of organs you take, and also the type of cells within that organ. All right. So why do we care about regeneration? We do care because regeneration is required, uh, and regeneration is one of the major challenges in plant genetic engineering. Even if we can transform a plant with whatever gene of interest we have, if it does not regenerate, then there is no point of doing transformation. So of course we care about regeneration. So what do we know so far? What we know so far is we can take any type of explant uh, and we can regenerate into an entire plant. Sources of explant for transformation regeneration can be anything: protoplast, root, hypocotyl, cotyledon, leaf, petiole, embryos, and any kind of explants we can take. So when we do after we do the transformation, what we do is we transform them to a callus induction media for the differentiation of the cells, and then we transform those calluses into the shoot induction media for the regeneration of the shoots. So after we transform these cells, these explants, after transformation into the callus induction media is rich in cytokinin. So cytokinin induces, so as soon as we transform these explants into the callus induction media, the first transcriptional programs that gets activated due to the cytokinin present in this media, the callus induction media, is the PLTs. PLT3, 5, and 7 gets activated. These are the transcription factors that gets activated, and they further activates other PLTs 1 and 2, which are very important for pluripotency acquisition in the plant cells. So PLT 1 and 2 gives pluripotency to the cells, and pluripotency is required for plant cells to gain totipotency, and these totipotent cells can regenerate. So PLT 1 and 2 play important role here. I'll come back to COC1 and COC2. And then when we transfer these calluses from shoot induction media, which are highly proliferating cells, into the shoot induction media that is rich in cytokinin, what happens is that we need, there comes, so PIN1 is another gene called PIN formed 1. So in callus induction media, which is rich in oxygen, now when we transform them into the cytokinin media, we need to get rid of most of the oxygen because we are transforming them into the cytokinin media. So this PIN1 formed is a effluxer protein, effluxer gene, that effluxes the excessive amount of oxygen from the cells, and then which is required for the shoot formation. If there is excessive oxygen in calluses, then it takes a different direction and forms root. So we wanted to get shoot. So that's why this gene PIN1 effluxes and then it regenerates. And then, sorry. And then comes and then two typical meristematic activity comes into play, Wuschel, clavata, and all the morphogenic regulators, and it regenerates a new de novo shoots like this one. COC1 and COC2 are also get activated by PLD 1, 2, and 3, but they take a different, so they also helps in de novo shoot formation through immunogenesis. So it takes a different path of regeneration into the shoot. It takes embryogenic pathway. So this is what we know so far. These are again the pseudoapical meristematic activity. So what we know so far is we have callus, PLTs come into play, PLTs, and it activates other PLTs, and PIN1, it effluxes the excessive oxygen, and Wuschel and clavata comes into picture, and it regenerates. What we don't know, so whatever we know, that is like a tip of an iceberg. Whatever we don't know is a lot there, where a single cell regenerates from a single cell to the whole plant. We don't know there's a lot of missing information here. However, there are some reports saying only some stress-associated genes helps in the regeneration, helps in navigating a single cell to the whole plant, and also the chromatin accessibility. It says that when we isolate the protoplast, chromatin accessibility increases, and that helps in the regeneration processes. So through this study, I'm trying to unlock or decode all the different molecular mechanisms involved in a single cell regeneration from protoplast to the whole plant. So how am I going to do that? So this is basically divided into two parts. One is the first one is wet lab part. So I will be isolating protoplast from tobacco, and then isolating nuclei from that tobacco, and run it into a single cell microfluidics, generate library, and then get the sequencing data. And the second part, which is a dry lab part, is basically the analysis of that data. After we get the sequencing results, we align the reads against the reference genome. We'll get the expression atlas. We'll get different cell clustering, and comes the identification of these clusters, and also the trajectory analysis or pseudotime analysis, and we'll land into candidate genes. So, and we'll say that okay, these genes are could be playing crucial role in the regeneration of single cell into a whole plant, and then further comes the validation of those candidate genes. So in this experiment, we have considered 6 different unique developmental stages. First one is the protoplast, immediately after isolating the protoplast. Second is after 5 days of incubation of the protoplast in the media. In 5 days, they look like this, dividing protoplast. And after 2 weeks, they will be massively dividing. This is called microcalli stage. And then when we transfer these into the callus induction media, they form calluses. When we transform it into the soot induction media, they form, as you can see here, scanning electron microscopy. These are the soot primordia. They form soot primordia at this stage, and eventually they regenerate. So we have considered these 6 different developmental stages for single cell transcriptomics. Basically, for each stages, we take the sample, we take for example, day 0, we isolate the protoplast and immediately isolate the nuclei. The white dots here, you can see those are the nucleus under microscope. And then we pass this nuclei from all the different stages through the single cell microfluidic system, where each individual nuclei is encapsulated into a gel bead, which has 10x bar-coated gel beads and an oil like this. So this is a gem, and your cell or your nuclei is connected with the barcode, and we prepare library from here and send for sequencing. So after we send it for sequencing, we get the first thing we see is the clustering of the cell. So for example, this AK1 is the sample number 1. So this is AK1, 2, 3, 4, 5, and 6. So this is number 1, day 0 sample, and each dot is a resemblance of one cell. If you can see this table here, so we were able to, so out of 10,000 cells you supply to the 10x microfluidic system, not all 10,000 you'll get high quality data from all the cells. So in this case, we got like, in all the samples, we got about 10,231 high quality nuclear cells. And their distribution based on different samples is like this here. And if we look at which sample is where, so this is day 0 cluster for protoplast, day 5 here, and 4, 5, and 6 is all here. So this is part of the, so they all have callus and they are kind of mixed together. And unfortunately, one of our samples got failed, so I don't have data for that particular sample. So this is a sample-wise distribution or clustering of the cells. And this is now those different samples are further divided into different clusters. And each of these clusters, we have 14 different clusters here. In each of these clusters represent a unique cell types that has been formed during a single cell navigating into a whole plant. And this figure here shows how the cells are divided in each of the clusters. All the clusters are here, how they are separated into different clusters and different across different samples. And this provides actually a good information how different samples are divided into or clustered into different clusters from different samples. Now, after we have these different clusters, for example, we have 14 different clusters here. Now, the challenge here is to identify what these different clusters are or what these different cell types are. And to identify these cell types or different clusters, we have different markers for each clusters. For example, for sample number 1, cluster number 6, there will be different markers based on their expression during the regeneration processes. And so we are still analyzing the data. We have not yet identified all the clusters, but how we are going to identify these clusters is an example is here. For example, this gene here is expressed in only, it is expressed, so this is expression here, 1 through 4. The more darker it is, the higher expression. So it is more expressed here in this cluster, and it is scattered in different other clusters. But when we look into that particular gene, we can see that okay, each of these dots represents the number of cells where this particular gene is expressed. So if you look at the sample number 1, which is AK1, most of the cells in sample number 1 is highly expressed there. And if we dig down into the cluster here, if you see the cluster number 6 here, which is right there. So this gene could be a marker gene for cluster number, to identify the cluster number 6, which is right there. So this is how we get the marker genes, and also based on previous literatures and bulk RNA sequencing data, we also can find different markers. Similarly, there is an example of second marker gene, which is expressed more over here, and that is sample number 2, and which is expressed in cluster number 11. So this could be another marker gene for identifying the cluster number 11, and so on and so forth. So this is how we identify all the clusters. And we have not yet identified all the clusters, and we are in the process of identifying the clusters and annotating them as giving them a particular cell type name. So moving on, after we identify the clusters, there is another thing called pseudotime or trajectory analysis. Pseudotime is basically how a protoplast or a single cell navigates through all the different stages to the stage where it forms the whole plant. So how it moves from the start to finish. So the color of these cells here, clusters or cells, is basically the age of the cell. If you look at this is the youngest age of the cell, and this is the oldest one. So it starts from right here and then goes all the way down here. So this is sample number 1, and it navigates to the second sample, which is AK2, and it goes and branches out from here. So branching out, and 1 goes to this direction. So what that means is whatever cells takes this direction, they terminate there. They cannot move away from there. So they could not regenerate. Their life ends there. But whatever cells goes from there and navigates through all the way to the sample number, sorry, 6, which is cluster number 12 there. Those are the cells starting from there, navigates through all these different stages and regenerates there. This is sample-wise pseudo-time trajectory analysis, whereas this is now cluster-wise. So it starts from there, which is AK1, and goes all the way and up to the sample number, sorry, cluster number 12, which is AK sample number AK6. So this gives a lot of information that how a single cell can navigate through this, and different genes are differentially expressed during this navigation or pseudotime. I'm going to show you some of the genes that are uniquely expressed during this navigation. If you look at these 4 different genes here, so here this is sample number 1, AK1. This is the golden color is AK2, and these 3 samples are 4, 5, and 6 here. So these particular genes, these 4 genes are highly expressed on the first day 0 data, where and then they slowly their expression goes down, and then later it goes up. So the pattern looks similar for these 4 genes, where their expression is very high in the initial stage, and then that slowly goes down and then goes up. Similarly, there are other set of genes which gets whose expression is higher in the second stage, or the sample number AK2, and then it was very low initially, and it rises up, and then it goes down again, and it rises up again. And the third type of set of genes are like their expression was very low in the beginning, and then that rises up towards the end of the pseudotime. If we look at that pseudotime picture, for example, here, so some of the genes have very low expression here, and then when the time flies, the expression goes up, whereas some of the genes have very highly expressed over there, and when it reaches to these stages, its expression goes down. So those are the very important genes, pseudotime genes, that could play important role in plant cell regeneration. So basically, we don't have a conclusion yet, but we are still working on the data, at least for the second project. In the future, we wanted to identify all these cell clusters and cell types, and land into some candidate genes and functionally validate those candidate genes. With this, I would like to thank Dr. Gunwant, my advisor, who has been supporting me since day 1. And he provided me with those challenging projects. And also I'd like to thank Dr. Lewis. He is my co-advisor. Dr. Inping Jiao and Dr. Boro are my committee members. And I would also like to thank all the members of my lab. And also I'll not forget to thank Lenin, who is a postdoc in Dr. Lewis' lab. He helped me with the single cell work. Thank you.
Thank you, Arjun, for amazing presentation. We have time for a few questions. If you have any questions, please. I have a few questions, but one of the questions is, as you already have your cascade and you are trying to do transformation with recalcitrant crops like soybean. So in your cascade, you have 3 different development regulators. Do you change the gene sequences, or like those developmental regulators? Are they specific to one crop only, or with another crop you use their own sequences?
No, they are all same. So for each any type of crop species, so in the figure there was like inducer and DR1, DR2, DR3. So what I make is I make one cascade with one DR only. And sometimes for recalcitrant species, for example, soybean, I have two DRs combined in one cascade. But no matter what kind of crop you want to test this cascade, you will not change the sequence. The DRs remains the same.
Is there a difference between a gene edited plant and a transgenic plant?
Yes, as well as no. Yes, because difference between gene edited and transgenic. Gene edited is where you target a particular gene and precisely knock out that gene and make it non-functional. Transgenesis is where you can overexpress a foreign gene, for example. You are not going to knock out that gene. But when we talk about gene editing, I think there is also transgenesis, because we not only provide the cascade with the guide that will target a particular gene, but also we provide it with the Cas9. So I think it has both uh, uh, transgenesis and gene editing.
Is there any question from the audience? First of all, it was a very good presentation.
Thank you.
I have a general question. In your first project, I have seen that you have improved some efficiency, like 5 to 12%. So what's the basis of that? How did you calculate that?
Counting the numbers. So if you inject like 100 plants, if you get gene edited shoot or a transgenic shoot from 12 plants, that is 12% efficiency. Basically, if you inject in multiple sites in one plant, that also counts. For example, if I inject 100 plants with 3 different sites, so that is 300 different sites of injection. But in my case, since it was only one point of injection, if I infect 100 plants, whatever number of plants get regenerated or gene edited, that is the frequency. Yeah.
Now, I would like to thank you, Arjun, again for your wonderful presentation.
Yeah.
So that's it. I would like to conclude the seminar series. I would like to thank Arjun again and Dr. Bhatt for allowing him to be the part of this seminar series.
Thank you.